right, I'd like to officially welcome all of you to the California Initiative to Advance Precision Medicine Symposium on Cancer Disparities Research. I'm Julianne McCall. I'm very proud to be the director of the initiative and serve as the director of precision medicine within the California Governor's Office of Planning and Research. I'm joined by my extraordinary staff, including Dr. David Reiner, who will be emceeing the remainder of this symposium. Before I pass off the microphone, I just want to thank the teams for the tremendous dedication that you've all shown. I remember May 1st, 2019, we gathered in the Governor's Office conference room to launch these projects, and the energy of the conversation and just the extraordinary hope shared across teams just gave me, enormous inspiration. And I'm very proud that we've made it to this stage, very proud as a state to have supported such world-class researchers and community partners trying to help change and update the paradigm of community-driven and patient-focused research in cancer. So with those very brief remarks, I'm going to pass it off now to Dr. David Reiner, Science Officer in the Governor's Office of Planning and Research.
Hi, everyone. It's a pleasure to be here with you. Thank you, Dr. McCall, for that introduction. My name is David Reiner. I'm a Science Officer, which means I oversee our demonstration research projects, including our 3 projects on cancer. And we're delighted to welcome you to this symposium on those projects. We have 3 presentations today, one on each of those 3 cancer projects. And I'll just get right into it. Excuse me, one more thing. After the presentations, we'll have a discussion, followed by kind of a closeout, different closing remarks. With that, I'll introduce our last speaker, Dr. Elad Ziv, who is a professor in the Department of Medicine at UCSF. His research focuses on breast cancer and other age-related diseases with a human population genetics and epidemiology approach, and with a particular focus in Latino and Latina communities, as well as African-American populations. Dr. Ziv received his MD from UCSF and completed his residency and fellowship in internal medicine and clinical epidemiology, also at UCSF. He is joined by members of his project team, including Dr. Susan Newhausen, Dr. Miriam Hernandez, and Alejandra Martinez. And with that, Dr. Ziv, take it away.
Thank you. Let me share my screen. You all see it?
Yes.
Great. Well, thank you for finding us, and thank you also for for inviting us and give us an opportunity to tell you what we have done in the last few years. Sorry. Nope. Something's going wrong. Okay, sorry. There we go. Okay. Try again. All right. So our mission was really to improve disparities, to improve the understanding of disparities in precision medicine. And we broke it down into several components. One is to improve genetic risk assessment, so hereditary breast cancer risk assessment. The second is to improve knowledge about genetic risk assessments among Latinas. And the third is to identify important cancer or somatic mutations in breast tumors among Hispanic Latina women. And I'll just jump right in and tell you, aim 1 was really kind of the hereditary cancer risk. We broke it down into 3 sub-aims. One is to analyze variants of uncertain significance in known breast cancer genes. The second sub-aim was to improve polygenic risk scores in breast cancer in Latina women, and the third was to increase knowledge about hereditary breast cancer among Latino populations. So in AIM 1A, the goal was to reclassify variants of uncertain significance. These come back more commonly in populations of mixed ancestry or non-European ancestry, just because there's less testing in these populations. And so we started with 4,200 cases and 4,300 controls approximately. This is the results of our first analysis. This is just a high level analysis. These are the odds ratios associated with breast cancer. So if the point here is on the right of the dotted line, it's increasing risk. And if the whiskers or these little bars cross the dotted line, it's not quite significant. And if the color is in the tan color or yellow, those are actually not just a little bit significant, but they're significant, adjusting for all of the known genes in the genome. And you can see for overall breast cancer, BRCA1 and BRCA2, which are obviously the well-known breast cancer susceptibility genes, and PALB2, which is also a well-known susceptibility gene, jumps out. And then we broke it down by ER-positive breast cancer and ER-negative breast cancer. And for ER-positive, the things that were really strong were CHEK2, which also is a very well-known ER-positive breast cancer gene. ATM, which is actually accepted ER-positive breast cancer gene, was actually not associated in our data, and we're still working out to try and figure that out. And then for ER-negative, estrogen receptor negative breast cancer, which is related to, not exactly overlapping, the triple negative that we just heard about that the UCSD team was working on, we saw BRCA1 and BRCA2 associated, but also this other gene called FANK-M, which is a member of the Fanconi anemia pathway and had been implicated in ER-negative breast cancer before our studies. But to our knowledge, this was the first time it's been identified after adjusting for all known susceptibility genes. And the association that we have found is stronger than has been reported in other populations. That report is a manuscript under review currently. We also managed to recruit 1,679 participants as of, I think, earlier this month. These are Latina women with breast cancer. They recruited City of Hope at UCSF from the clinics at both sites, and also from the California Cancer Registry. This was really a tremendous effort by Veronica Tejeda and Anna Fernandez at UCSF, who really did all of the work here. It was a remarkable effort during COVID. We were kind of, unfortunately, having to do a lot of this early on by mail and so on, but they managed to both get started during COVID and work by mail and get saliva samples sent by mail. And then when things reopened, they managed to go back into the clinic and recruit a lot more. So we were very proud of the work that they did. We are now sequencing. We actually completed sequencing, and we're now getting close to done with analysis of 70 genes that are associated with breast cancer, including all the genes that you see on this figure and a few others, and about 2,500 cases and about 200 controls. And we're going to jointly analyze that with all of us data set in a complete data set that would be about 6,700 cases and over 30,000 controls. And we hope that there will be not only much better variant of uncertain significance classification, but also possibly genes that may be contributing more in this population that have not been well recognized until today. So I would say that AIM 1A gotten pretty much all of we had set out to do, and perhaps a little bit more, in the sense that we're beginning to potentially identify new genes that are important in this population. In AIM 1B, the deal was to improve polygenic risk score for breast cancer in Latinas. Currently, we are using genome-wide data from 5,700. So this is array-based data. It's a different technology than the sequencing that I just showed you in the previous slide, although many of the samples overlap. And we have data from 5,700 cases and almost 13,000 controls. The idea for this grant was we were going to analyze what's called the confluence data set, which would have 24,000 Hispanic Latina women and over 24,000 controls. The confluence was not going to be, the genotyping wasn't sponsored by this project. It's done at NCI and sponsored by NCI. And it's just taken longer. And so we will be analyzing that at a future date. But actually, Laura Fairman's team has been doing a lot of this analysis. This is the data in terms of the ancestry of the populations, and we've included both women from the US and also women from various studies recruited in Mexico. And what you can see is this is the genetic ancestry. The blue is the European ancestry. The green is the African ancestry. The purple is the East Asian ancestry, which is there a little bit higher in Peru, kind of as is known, but overall very low. And then the red is the indigenous American ancestry. And that's the two components are really the indigenous and the European. And the indigenous is highest in Peru and higher in Mexico than in the US. And so this is kind of the summary of the work that Laura has been spearheading. And these are polygenic risk scores with various SNP panels that we've included. So I'm going to have to move the window so I can actually see. But the panel on the 3,500 SNP panel is sort of the latest European polygenic risk score. And what you can see is that works well in the participants with the lowest indigenous ancestry and consequently highest European ancestry. So if you move to the right, the polygenic risk score is more predictive. But what we had also done before is included SNPs that we had identified. And when you add those back in, what you can see is really a lot better results in all of the populations, but particularly the highest indigenous ancestry populations really move much farther in terms of increased prediction of the model. And so what we're hoping is that with the confluence data coming in, that we'll be able to further improve, particularly in this population, because we'll be able to get better estimates for those rare variants that are specific to the indigenous population. The second part of the work is the educational component that Laura Fairman co-ran with the community partners, Visión y Compromiso and Promotion of Better Health. And what they did was really remarkable work, also having to kind of work around COVID. So they did took promotores who were already working with the population, but needed enough genetics knowledge to be able to become navigators for women who are concerned about their risk, or even just educating women about their risk. And so they did the training over Zoom. They did surveys and actually evaluated how well the educational sessions went. They've already published actually several publications. These are at the bottom. And these are the results. So after the promotores were educated, they then went out into the community virtually initially, and then in person, and did outreach and these training sessions. And the training sessions have reached now over 1,900 women. The mean age was about 44, very low English fluency in this. So the sessions were pretty much all conducted in Spanish. Their large fraction of the women did not have health insurance. and actually the almost majority had public health insurance, and mostly didn't actually know about genetic testing. They were able to really reach out broadly in Northern and Southern California to do these training sessions, and found that about 7% have very strong family history, who they are now navigating into clinical testing, genetic testing. And this is the team, and also supported in part by UC Davis has a initiative called Lucha, which is really also doing community education around cancer. AIM-2 is a tumor mutational profiling aim, and in the first part we were doing our own tumor sequencing, matched germline somatic tumor sequencing to identify variants that actually, genes that are not recognized as potentially genes that are not recognized as cancer mutated genes, and they're not part of panels. And then in AIM-2B, where we're going to look to see in a much larger data set with Foundation Medicine if known recurrently somatically mutated genes are more commonly mutated in different ancestry populations. And I'll go talk about that now. So the first part of AIM-2A is we analyzed about 146 tumor normal pairs, and this is now published. What we found was that this is the somatic mutational profiles, so not necessarily looking at any particular gene, whether it's mutated, but basically just the pattern of mutations. And from that, you can often learn about what's driving the tumor. So the most common signature that is in these tumors, and it's also in tumors from other populations, is really a signature that's associated with older age. That's not surprising. Another common pair of signatures is signature 2 and 13. And that's actually a well-recognized signature for something called APOBEC. And there's a common germline APOBEC variant that's actually more common in both Asians and Latinas, and is associated with this signature, although the signature is seen in all populations. So we saw that. We also saw what's called signature 3, which is more commonly seen in the basal subtype of breast cancer, and is associated with defects in homologous recombination DNA repair. So that some of these tumors have BRCA1, BRCA2 mutations and other mutations that abolish their activity to repair double-stranded DNA breaks. And then we also saw this signature 16, which is not something that's been reported in breast cancer in other populations. And Ding, who led this work, actually also saw that a prior publication of a sequencing study in Mexico also saw this signature more commonly than has been reported in non-Hispanic white populations. And what was interesting is that this new signature is predominantly seen in the luminal subtype of breast cancer, so not the basal or triple negative breast cancer subtype. And then this is, I would say, very preliminary data that Ding has put together. This is results from 437 women who were sequenced as part of the City of Hope clinical panel. So we had the benefit of working with that as well. And we found that TP53 was the predominant gene. So this is actually TP53 is a well-known gene for all cancers, but it's a little more commonly mutated in the Latina women from City of Hope Clinic than the non-Hispanic white women from City of Hope Clinic. So it's associated more actually with TP53 is much more common in basal or triple negative breast cancer. And so that's something we're analyzing very carefully now. And one other gene that sort of came to our attention is this gene called CTCF. It's a known transcriptional repressor. It's associated with mutations in other tumor types, but not with breast in non-Hispanic white populations. And it is clearly recurrently mutated in this population. Not common, about 3%, but it's clearly playing some role. So this is the analysis now in 40,000 breast tumors with from Foundation Medicine. So Foundation Medicine is a company that's basically sequencing tumors for clinical diagnostic purposes. So they're not doing whole exome sequencing, they're doing more targeted panel sequencing. So that doesn't allow us to look broadly across the genome, but it allows us to look at the known breast cancer drivers and which are more common in a much larger data set. And because they are sequencing everybody, we could look across different ancestries. So we looked at both at African and Asian and Indigenous American ancestry. And these are the results from African ancestry. And what we saw here was that these are copy number aberrations. What we saw is actually just generally copy number aberrations were more common. Copy number amplifications, excuse me, are more common in women with African ancestry. These are both copy number amplifications that's associated with ER-negative breast cancer, which is more common in African ancestry, but also copy number amplifications in red that are associated with ER-positive breast cancer are more common in this population, which is a little surprising. And I think it's underlying the fact that DNA amplifications in general are more common in women with African ancestry. And then we saw PIK3CA is less common in women of African ancestry, but GATA3 is more common. And that's PIK3CA was known before the study. GATA3 is not actually known as a new finding. And then we also looked at the same analysis with Asian ancestry. And there are several genes on the right of the panel that are more common both with Asian and Indigenous American ancestry. One of them I just want to highlight is EGFR. So this is a gene that's not considered a common breast cancer driver, but it was present in a small number of breast tumors, but it's also well known as a great area of difference in by ancestry in lung cancer. So EGFR is very common in lung cancer and very common in East Asian populations and to some degree Indigenous American populations with lung cancer, much lower in European ancestry with lung cancer. And what we've identified for the first time is that that same pattern holds with breast cancer. So it's overall much less common in all populations, but there's a fourfold difference in lung cancer, and that same fourfold difference exists in breast cancer. And the second panel here, the lollipop plot, just demonstrates that this is true across the gene for all of the known recurrent mutations. And I think this is just sort of our summary slides, and we are working actively to try and maintain the research. Dr. Feherman got a new study to study the genetics of HER2 amplified tumors in Latina women. And Susan and I are working on a grant to try and do more of the sequencing that we mentioned. And there are other grants that are sort of being resubmitted and under review. I think I'll stop there. And I just want to say this was a tremendous team effort by a lot of talented and devoted people, and thank them for that effort. Then I'll stop.
Thanks, Dr. Ziv. We have time for one question for Dr. Ziv. Anyone?
So I have two quick questions. Number one is when you were doing the germline analysis, if you were to divide them up into the young and old, were you going to see some signals become stronger? Because obviously young cancer incidence is in the rise. And then the second question I had was with regard to the somatic analysis that you did. Were these treatment naive samples, or were they sort of post-treated samples?
Right. So I think all of the genetic variants were more common in younger women. We did that, I think, at one point. It didn't sort of jump out at us super strongly. But what I think we saw that was much stronger was that, and I should say, in general, this population is quite young, both the cases and the controls. So, you know, I don't think we have a lot of really old women, but what jumped at us really was more the ER positive, ER negative results. When we finally saw those results, it was almost like, you know, kind of the light had been turned on in the room, and it turns in the sense that, you know, FANK-M really jumped out at us. We went back and read the literature about FANK-M, and actually that's what's always been the case. It's an ER negative or triple negative breast cancer risk factor. We also saw there's a couple of genes I didn't include in there that are good candidates that are potentially novel, and also those came up really much more clearly with the subtype. So I guess based on that, I feel subtype is a little bit more informative than age.
Yeah, we should chat after this meeting, but I mean, I think that even dividing up the groups into ER positive and negative will, you know, we'll be able to kind of further subset that into a more sort of homogeneous population.
Yes. The problem is I think we have limited, like we have HER2 on most, but not everyone, because some of these are really historic samples before HER2 is commonly done. And I don't think we have like a lot of other things, like these are all immunohistochemistry kind of, I mean, the subset, so there's about 700 that we have tumor normal on. I showed 146 initial and the 437 that were just more kind of homogeneous from the City of Hope, done in the same batch. But we're putting together a data set with 750, and those are tumor normal. We can do a lot more germline versus like what's going on in the tumor. And to answer your question, I think the second question was, it was tumor normal whole exome. Is that the question?
No, the treatment.
He asked how many were tumor naive or treatment naive.
Oh, they're all treatment naive. There's a few.
No, that's not true. The 136 are all treatment naive, and the 37, they're not all. We're still gathering.
Okay. I think sorry about that. I meant to show the treatment naive subset. But
you did. That was 146.
Right. But the 400. Okay. Anyway, the final analysis will be, I think, sort of for treatment naive, because I think that it's just harder to interpret the non-treatment naive participants.
Thank you.
So
go ahead, Isabel.
I'm going to say, Elad, can I add? Because I think that while we're looking at this hard science, and I think it's excellent and exciting for our community to have some real look at the issues, I think one of the things that should be pointed out that I was excited about, because Laura and I worked on creating the hereditary breast cancer toolkit that was then tested by Miriam and the promotoras very critically. And one of the things you didn't mention that I thought was important, out of those 1,900 women, 500 were identified as lacking cancer screening, colorectal, breast, and cervical. And this was where we're talking about the early intervention piece, because we know that the smaller percentage are, of course, women with genetic risk. And so that is why we like the combination to everybody of the community engagement piece, because I think we can help identify through the education and awareness the early interventions that need to be done and that are still going unserved, as well as looking at these risk factors so that we can cover the whole thing and get some really impressive numbers and address some of the disparities. I'm super excited about what they were able to learn using, employing our hereditary breast cancer toolkit, and see it as a very valuable added piece for community agencies to be able to start to inform communities at scale about hereditary breast cancer and still identify those patients who need regular screening. Thanks, Elad.
And the other thing, sorry, and maybe Alejandra, the Promoters for Better Health can comment, was you know, COVID on some level ended up being good because they were able to reach so many more women by doing things through Zoom rather than trying to have in-person meetings or education meetings.
Great, thank you so much.
I just want to, sorry,
go ahead.
I want to echo one other thing for Isabel is, I think that this is, thank you for saying this. I think that, you know, disparities is a really complicated thing, and there's disparity. I talked a lot about the disparities at sort of the cutting edge, precision medicine and sequencing and blah, blah, blah. But a big part of disparities is just like doing like the basic stuff, you know, like that we all know sort of works, and kind of getting that actually done in an effective way, I think is really important, and I'm glad you raised that.
Well, I guess I can just conclude then. I'm pleased to see that all of our projects brought aboard community-based organizations and/or community engagement in order to show that across this continuum, the community is really helpful in jumpstarting the work. So thank you very much. I appreciate it. And I loved all the presentations for what I understood for us non-science types.
Dr. Ramos, were you going to say something before I move on? Go ahead.
Yes, thank you. And I think the point was just brought up for us non-science type. So you all are brilliant in terms of really knowing the science and distilling the precision medicine. But I encourage us to now translate that into simple terms for those of us who are not at the bench, for those of us who are not dealing with and I love Dr. Ziv's explanation of you know, is it significant, not significant, and so just simplifying it to have others understand, and then not only what the science is showing, but more importantly the value, because oftentimes we tend to, and I can tell you medical speak, we tend to speak in jargon because this is what we know, this is what we do. But I think we need to now simplify it so that others can understand and see the value. And then you're going to have more advocates to say, this is critically important, this is what we need to do. But I was very impressed with the science, and you took me back to my research years in when I had to do all of the tissue culture and all of the basic science. So nice presentations.
Does anyone else have anything to add before I move on? Great. So I just wanted to thank all our speakers, Dr. Patel, who was able to just join us, Drs. Kim and Tamayo, and Dr. Ziv, as well as every member of their project team and community partners, as well as all the participants and patients and families that were engaged in the research. I also wanted to take a minute to acknowledge CIAPM's former program officer and co-director, Dr. Shannon Muir, for her talented oversight of the Cancer Disparities portfolio for almost the whole duration, except for the last few months. I'd like to thank our advisors, Surgeon General Dr. Ramos, Mr. Ron, as well as our Advisory Council Chair, Dr. Lajan Sher, for joining the symposium, and we'll hear remarks from them towards the end of our discussion session today. So that concludes the recorded presentation session for today. So I'll stop the recording and then pass the mic to Julianne to open the discussion session.
Thank you so much, David. And to echo the expression of gratitude, like I mentioned in my opening remarks, it's been just a true privilege to allocate state funds uniquely to cancer disparities research projects with a precision medicine lens The entire goal of our initiative is to demonstrate the promise and potential of precision medicine. And we really look forward to presenting an evaluation report to the legislature through a process that I'll mention toward the end of our symposium here in the final 40 minutes. But before we conclude,